thank you, DG, for the handling of the previous session, which I think just lays the foundation of what we are about to get into. So as it has been said, we have started to get into this fight against this enemy since last year. And one of the art of war is that you never fight with your eyes closed. So you always have to know and be very clear about who is your enemy and how your enemy is advancing. And one of the ways of doing that is actually to be able to do genomic surveillance, so that in fighting for epidemics such as we're doing now, you know the kind of the makeup of the enemy that you're doing. And this is really where the whole work around the sequencing and the identification of variants comes in. And this is really quite crucial, because without this knowledge, you can end up with all billions of rands being invested into vaccine manufacturing, while you are actually just developing water or salt solutions because they will be ineffective. So in this area, I'm just pleased that we now joined with one of the top scientists, not only top in South Africa, but globally known, Professor Tulio de Oliveira, who is going to take us through some of the work that they've done in CRISP, which work has informed largely a whole lot of work that is happening globally. And I really just want to be thankful that the investment that we make as a country in science and technology development are actually also playing a key role in the global ecosystem of technology and innovation development. So I'm going to, at this point in time, without any waste of time, hand over to Professor de Oliveira to take us through what is it that we talk about when we talk about variants, and what are their relevance in this fight that we have embarked on. Prof, I'm going to hand over to you, and we hope that as people are going to keep on putting on their questions and comments, we'll also find a way of following up and dealing with that. Over to you, Prof.
Yeah. Good afternoon, colleagues and viewers. Yeah, good afternoon, Honorable Minister, DG and DDGs. Yeah. So what I have to share for you, yeah, it is something that I think that South Africa can begin to be proud of. Yeah. It is our what Mbomeni, the DDG of Innovation highlight, and I believe also our Honorable Mr. Zimandle, how we end up by setting up this big collaborative network, yeah, become almost an example to the world on how to use this very advanced technology, genomic surveillance, yeah, to not only discover new variants, but how to feed that back for evaluation of vaccine and development of vaccine, which I believe it is a very crucial step that South Africa will be taking in the next few months and few years. Yeah, so it's very important. I'm presenting that sometime. I'm not presenting for myself. I'm presenting for a large number of groups and individuals, which involve a lot of our universities, yeah, and the national laboratories, yeah. So I like always to start by highlighting that this is not the work of one scientist or one group or just CRISP, but this is the work, it's very consistent work over the last year. We are 11 months doing this week by week. And what we have done quite early is with investment from the Department of Science and Innovation and the South African Medical Research Council, we set up these 5 sequencing facilities, one at CRISP, University of the Free State, Stellenbosch, University of Cape Town, and at the National Institute of Communicable Disease. More important, each of them, it is hands in hands with the NHLS lab, which allow us to do this genomic surveillance very quick and very accurate and feedback data, not only for the scientific community, but for the public health officials, such as the Department of Health, continentally, the Africa CDC, and internationally of the World Health Organization. Yeah. So our network was, the idea of our network was initially published in the Lancet Microbe with both Kainim Somi and colleague Melisana as the first authors, just to highlighting the importance of doing that together with the NHLS. Yeah. And it is this work that we have been done has become like really hail around the world there. So for example, it was covered on over 500 articles, a lot of them in the BRICS country or the G20s or in the Latin America, really highlighting how a developing country like South Africa can start implementing this kind of technology and emerging that. I always like to put this, it came a number of times now, it's 3 or 4 times in the front of New York Times, yeah, about our work. But this one was special because that's the same day that the Trump was impeached or was kicked out of Twitter. So it was a very competitive day from the front page of the newspaper. Yeah. Last week, it came by the director of Francis Collins of the National Institute of Health. That's the biggest National Institute of Health funder, showing how South Africa has shown the power of genomic surveillance in the COVID pandemic. Yeah. So how we did that? So what we did, we start very early last year, like end of February, yeah, and we start every week working with our national labs, getting samples, moving that to genomics facility and sequencing the virus in real time, yeah. When I have white here, it's not that I don't have data, it's the opposite. Normally I have 20, 30 or 40 lineages, mostly originated from Europe. And then around May, we start getting our own lineages in South Africa, which 3 of them were kind of co-dominating the infection, and they get numbers in the classification system, yeah. But then what we have is the appearance of this other one, this 501v2 that appeared in middle of November, and very quick start dominating, yeah, and dominate and displace all the variants and all the lineages, yeah. And that was worrying how one variant start transmitting so quick and displacing all of the ones circulating in the country, yeah. We can map that in both space and time. What we know is that this big amplification event of this variant happened in the Nelson Mandela Bay in the Eastern Cape, yeah. That is what we think that the variant emerged and transmitted southwest to Cape Town and northeast to KwaZulu-Natal. It's very important to notice that we don't know if this variant, it really come from South Africa or was external introduction, yeah. What we know is that was amplified. It follow a lot our corridor of both, especially national roads, yeah, and very quick start dominating all of the infections in the whole country, yeah. What we also know is that this variant has many different mutations, and I'm not going to get in big details, but what we know is that around like 23 mutations within close to 20 mutations are amino acid change mutations. What does it mean? It means that a mutation that changes the structure of the virus. And we're particularly focused on these mutations that are in what we call the spike gene, because that's the main protein that will bind to the cell to enter. And that's the main protein that's also is going to be attacked by antibodies. And the variant that we discovered in South Africa has many mutations, around 9 mutations, and 3 of them in the most important region of the protein, which is in red here, the receptor binding domain, yeah. What we know that now we have detected the variant. We also know that in South Africa, it has been very crucial on the trial development of the vaccine. So what we did is to partner with 2 colleagues, yeah, or 3 colleagues. One is Shabir Mahdi that was run the AstraZeneca trial, but also with this fantastic work done by Penny Moore at the NICD and Alex Siegel at the Africa Health Research Institute. What we know, we have the variants, and what we can do, we can expose the plasma of vaccinated individuals to the original strain, to the one with the mutations only in the receptor binding domain, and to the variant, which also get the name of B1351. And what we see is that you decrease the effect of antibodies from the vaccines and the AstraZeneca on individuals that are vaccinated. This is very important, mild disease. This trial was a smaller trial, didn't have anyone severe disease. And we did that with colleagues on both two different techniques. One of them is what you call the pseudovirus, very advanced techniques, and the other one is in the live virus. Yeah, and this is the most advanced one, what you call the global standard, and we see this big decrease of neutralization. Yeah, this kind of data is very important to feedback straight to vaccine manufacturers, but also to national governments. Yeah. So what we did, we have set up in South Africa with colleagues from all major universities in the country, this system that we can go from genotype, very quick genomic surveillance, identifying variants, but see what's the effect of variants on that on vaccines. Yeah. And I'm just listing here some of the main vaccines that has been produced, such as the Johnson & Johnson, the Pfizer, the Moderna, the Sputnik, what's also go by the name by Gamaleya, the Novavax, Sinovac, that's the Chinese one, and Sinovac. And what we are doing with colleagues in South Africa, using both a pseudovirus and live virus, but around the world, is see the effective of the variants on decreasing the fold of effect of the vaccines. And what we say that they slightly decrease the fold of effective of vaccines, yeah, but some of them big more than the others, yeah. And then if you put that information together with the vaccines that have been evaluated in South Africa so far, we have the Johnson & Johnson that came the results. Why that's very important to run clinical trials in the country, so you can know the effect of the vaccine, not only on the lab, but also on clinical efficacy on mild, moderate, severe, and hospitalized and severe. So the Johnson & Johnson had a very strong efficacy on the, especially avoid the hospitalization severe of 85%. And now I believe that all the other vaccines as being rolled is gonna be evaluating the same. And that's one of the reasons why the national government decided to start rolling out the Johnson & Johnson to healthcare workers, because this one we know that work very well in our variants. We do expect most of them to do well with clinical efficacy, and this data will be accumulating the next few weeks. So the important take-home message is that now we have a very advanced system in South Africa to evaluate the efficacy of the vaccines very, very quick. Yeah. So just because my topic of the presentation is not just to focus on the variant that we have discovered in South Africa, so I just want to finalize with a few slides of all the main other variants in the world. So the one that has been identified in South Africa, again, we don't know if originated in South Africa. We know that went for a massive amplification event in South Africa. Now it's found in 46 countries around the world. Yeah. So these are countries that have reported that they have the variant, yeah, and 37 of those not only reported, but they made the genome available, so we could compare. And so that involved most of our neighboring countries, yeah. We are still missing data from Zimbabwe and Botswana and Angola, but we are being produced at the moment. So a lot of them, but also this variant being found in North America, Europe, and Asia and Australia, okay? They have also another variant that it is go by the name of B.1.7 or the 501YV1. That's the major, the first variant that's spreading widely with this mutation that also share with the South Africa. And that was first identified in the UK. And this one have been identified in 93 countries. Yeah, so much bigger penetration, both in North America, Central America, Latin America, but also Asia and Europe at very high level. And that we also have been found in South Africa. Yeah. What it means is the one that have been identified in the UK more transmissible than the South African one? No, both of them seem to be more transmissible than the normal. But what tells us is that the UK is much more connected to the world. And that's highlighted this effect that we have to treat this pandemic as a global response. Otherwise, variants that emerge will spread very, very fast. The third variant, that is what's called variant of concern, that's the 501Y.V3, the third major variant that has the mutation 501, which also go by the name of P1, was firstly identified by travelers in Japan. When they arrived, they are quarantined and they did the DNA test, but then was linked back to Brazil, especially in Manaus, that's the capital of the Amazon state. That one is spreading also and has spread into 31 countries, again, neighboring countries and entering Mexico, North America, and Europe. Yeah. At the moment, I'm part of what you call the World Health Organization Evolution Working Group. We keep an eye on these new variants. Yeah, we have another 2 in our watch list. One of them has been firstly identified in Uganda and has spread to Kenya and also to Rwanda, but begin to spread around the world. And the other one have firstly been identified in the UK, similar to the one in Japan, but was linked to Nigeria. So that's quite interesting. So potentially we have 3 variants, 2 of them in the watch list that has been emerged or amplified in Africa. And that's very important to continuous genomic surveillance in Africa and scale vaccination. Yeah. So what are doing that? And our Minister of Health highlighted that. We set up a number of labs across the continent. Yeah, in red it is what you call a regional lab, and in blue it is what you call a specialized lab. So what we have now, we have 12 labs across the continent that has extra capacity to provide genomic surveillance to across the continent. Yeah. So what we are doing is really to establish and supporting other countries to do that in real time. So for example, in CRISP, we are now helping Angola, Zimbabwe, Botswana, Mozambique, and Malawi. Yeah. The NICD is working close with Mauritius and Namibia and across other countries. So what we are doing is scaling this technology to help to do that across the whole continent. So just to conclude, genomic surveillance is a critical component of the epidemic response. Yeah, I haven't mentioned to you, but that's how people they develop the diagnostics. That's how they develop the vaccines, was based on the genome. In South Africa, we are very fortunate to get this strong support and vision from the Department of Science and Innovation, the MRC, to set a very comprehensive genomic surveillance program, which detects our variant very quick, within weeks of emerging. This variant has multiple mutations at key spikes sites in the main protein of the virus. We call that the spike protein, which decrease the neutralization of antibodies. The variant decreases the efficacy of certain vaccines, so it's very important, and that's why we have selected the Johnson & Johnson to start, and we are currently evaluating the AstraZeneca, and we'll be evaluating the Pfizer. But what is very important and highlighted by our minister is that now we are building the knowledge in the country, yeah, to really not only evaluate, but to design vaccines, yeah. So again, this is not the work of only my group, yeah, this is the work of hundreds of scientists across the country that came together in a network to try to respond quick to this pandemic. Yeah, thank you very much, DDG, and I pass back to you.
Thank you so much.